His brother's name was David, and what Frank, 68, remembers most clearly about that phone call, the one where David told him he was stage 2, caught just in time, is that his first thought wasn't about his brother. It was, when was my last colonoscopy? 3 weeks later, Frank is sitting in a gastroenterologist's office in Cleveland. His colonoscopy comes back mostly reassuring. One small sessile serrated polyp, removed on the spot. Nothing to panic about, the doctor says. Watch your diet, reduce red meat, eat more fiber, come back in 3 years. 3 years. Frank does what most people do. He buys psyllium husk fiber and starts stirring it into his orange juice. He adds a salad to dinner, cuts back on steak. 6 months later, he quietly stops thinking about it. The polyp is gone. The follow-up is scheduled. What his gastroenterologist didn't tell him, not out of negligence, but because research moves slowly from journals into 12-minute appointments, is that eat more fiber is the right instinct with the wrong specificity. There is a particular type of carbohydrate, prepared in a very specific way, that does something inside the colon that no amount of psyllium can. Something researchers at Vanderbilt University discovered while solving a paradox that had puzzled the field for years. Something that, and I'll come back to this, might change what Frank's 3-year window actually means. Before we continue, I'm Dr. Becker, health researcher and science translator, not a practicing clinician. What I'm sharing is grounded in peer-reviewed research. It is not a substitute for medical care. If you have a history of polyps, colorectal cancer, inflammatory bowel disease, or are under active treatment, work with your physician before changing your diet based on this video. This is educational information. Now let's get into it. The food is resistant starch, not an exotic supplement, a category of carbohydrate found in potatoes, rice, lentils, chickpeas, and green bananas, prepared in a specific way, because the preparation changes everything. A hot potato and a cold potato, cooked from the same batch, have fundamentally different effects on your colon biology. The cold one contains 3 to 5 times more resistant starch. The hot one behaves essentially like sugar once it hits your small intestine. Today, the cook and cool secret and why it works at the molecular level. The Africa study that showed colon cancer risk markers reversing in 14 days. The mechanism by which cancer cells are selectively starved. The way that same molecule repairs DNA by unlocking genes cancer had been trying to shut down. And the mistake that destroys all of this before it reaches your colon. What to do right now? Boil or steam potatoes tonight. Refrigerate them overnight. Eat them cold or gently warm tomorrow. Add a green banana to your next smoothie. Eat half a cup of lentils or chickpeas most days. Those are the foundations. Here's why they work. Not all starch behaves the same inside your body. When you cook a potato and eat it hot, the starch gelatinizes, unfolds into a structure your digestive enzymes break down rapidly and absorb as glucose. Your blood sugar rises. Almost none of it reaches the colon. But when that same potato cools, the starch recrystallizes into a structure called retrograde resistant starch, RS3. This form resists your digestive enzymes completely. Instead of being absorbed in the small intestine, it travels intact to the colon, where your gut bacteria are waiting. Do this daily. Boil or steam 1 to 2 medium potatoes. Refrigerate overnight. Eat cold or warm to no more than 130 degrees Fahrenheit. Above that temperature, the RS3 structure collapses back into digestible starch. 1 medium cooled potato gives you roughly 3 to 5 grams of resistant starch. Same potato, eaten hot, under 1 gram. Dorothy, 71, thought potato salad was a summer thing. Now it's year-round. A small bowl with olive oil and apple cider vinegar in the fridge, ready at lunch most days. She started because she read about longevity researchers, noticing that communities eating day-old refrigerated rice consistently showed lower rates of colorectal disease than those eating freshly cooked rice. Nobody called it resistant starch in those traditions. They were just eating leftovers. A 2019 crossover trial in Cell Host and Microbe, led by researchers at the University of Michigan, followed 59 healthy adults alternating between high-resistance starch and controlled diets. Basically, Consistent resistant starch intake selectively amplified exactly the bacteria responsible for producing the protective molecule your colon runs on, measurably within 3 to 6 weeks. That molecule is butyrate. Everything that follows is about what butyrate does. One practical note: if you've been eating low fiber for years, your first week will include gas and bloating. That discomfort is evidence the right fermentation is starting. Begin with one small serving and build over 2 to 4 weeks. The bacteria that make butyrate, Fecalibacterium prausnitzi, Roseburia intestinalis, Eubacterium rectale, are now called biomarkers of gut health. Their absence correlates with Crohn's disease, colorectal cancer, and metabolic dysfunction, and they decline. Research from University College Cork, published in Nature in 2012, tracked 178 elderly Irish adults and found the microbiome of people in long-term care eating processed food was dramatically more inflammatory than those eating varied diets. Same age, different diets, different colon environments. By age 65, estimates suggest 30 to 40% of the butyrate-producing bacterial population most people had at 30 has declined. Resistant starch is targeted refeeding for a depleted system. The question this opens: what exactly does butyrate do when levels are restored? The answer from Vanderbilt, when they figured it out in 2012, is where this gets genuinely surprising. Before the Warburg paradox, the Africa study. Because this answers the question most people are afraid to ask: is it too late to change anything? Add a green banana to your smoothie. One medium banana, green and firm, not yellow ripe. 6 to 9 grams of resistant starch in a raw granular form called RS2, which doesn't require cook and cool because the starch is already in its resistant crystalline structure. As the banana ripens, RS2 converts to simple sugar. The green banana giving you 7 grams becomes a ripe banana with less than half a gram. Buy bananas green, use them before they yellow. Pair that with half a cup of lentils or chickpeas at lunch or dinner, 2 to 5 grams of resistant starch each. Combined with cooled potatoes and a green banana, you're in the 15 to 20 grams daily range, where the published research starts showing meaningful biological shifts. Now the Africa study. I want to linger on the methodology for a moment, because the design here is more rigorous than the summary usually conveys. Dr. Stephen O'Keefe and colleagues at the University of Pittsburgh, working with researchers at the University of KwaZulu-Natal, recruited 20 African Americans and 20 rural South Africans. Not for a survey. They controlled every meal and had the two groups completely exchange diets for 2 weeks, bidirectional. At the start and end of each period, colonoscopy and mucosal biopsies, actual tissue samples from the colon wall, before and after. Published in Nature Communications in 2015. After 14 days on a high-fiber, high-resistant starch diet similar to the rural South African pattern, the American participants showed a measurable drop in mucosal proliferation, a direct cellular marker of cancer risk, and a significant reduction in secondary bile acids, a known cancer promoter in the colonic environment. In plain terms, their colon cancer risk profile measurably improved in 2 weeks. The rural Africans on a Western diet for the same period showed the reverse. Diet, not genetics, is the primary driver of the dramatic colon cancer rate differences between populations, and it starts reversing in 14 days. Colon cancer takes 10 to 15 years to develop, from first mutation to detectable tumor. By age 55, most people have microscopic precancerous cells already present. Those cells either receive the signal to self-destruct or they are allowed to compound mutations. What Frank does between appointments isn't passive waiting. It's 3 years of biological momentum going somewhere specific. Here's the paradox Vanderbilt spent years trying to solve. The foods remain the same: cool starches, green bananas, legumes. Add lightly steamed broccoli 4 or more times a week. Chop it first and let it rest 10 minutes before steaming. That window allows an enzyme called myrosinase to activate sulforaphane, which operates on a parallel protective pathway we'll connect shortly. Now the paradox. Butyrate should theoretically feed cancer cells the same way it feeds healthy ones. But colon cancer cells have rewired their metabolism to run almost exclusively on glucose through glycolysis, even when oxygen is available. This is the Warburg effect. It helps cancer cells grow fast, but it makes them metabolically blind to butyrate. Actually, let me back up for a second. Healthy colonocytes run on butyrate as their primary fuel, 70% of their energy. They absorb it, burn it in the mitochondria, and it's consumed entirely, never accumulates. But a cancer cell running on glucose can't efficiently oxidize butyrate, so it enters the cancer cell and stays, builds up. When concentrations get high enough, it migrates into the cell nucleus and inhibits a family of enzymes called HDACs. This triggers apoptosis, programmed cell death. The healthy colonocyte next door thrives on the same molecule. The cancer cell survival adaptation becomes the mechanism of its death when butyrate is present. Donoghue and colleagues confirmed this at Vanderbilt in Molecular Cell in 2012, including using germ-free mice with no gut bacteria at all. When butyrate-producing bacteria were reintroduced specifically, the selective cancer cell effect returned. Basically, your bacteria are exploiting a flaw in cancer's own metabolic design, a flaw that only becomes lethal when they have resistant starch to ferment. Remember what 3 years means for Frank. 3 years of either maintaining butyrate levels high enough to sustain that hostile environment, or not. This is the part I found hardest to put down when I first read it. Same protocol, cooled starches, green banana, legumes, broccoli. Add 2 tablespoons of raw ground flaxseed to yogurt or a smoothie daily. Ground, not whole. And 2 ounces of raw walnuts most days. Your DNA is wound around proteins called histones. When enzymes called HDACs, histone deacetylases, remove acetyl groups from those histones, the DNA coils tightly and the genes underneath go silent. Can't be read. Cancer cells use this strategically. By ramping up HDAC activity, they suppress tumor suppressor genes, the genes that say stop dividing, fix this damage, initiate cell death if this mutation can't be repaired. These include P21, P53 responsive repair genes, and BRCA pathway components. Butyrate blocks HDACs. When HDAC activity is inhibited, histones stay acetylated, chromatin stays open, and previously silenced tumor suppressor genes begin expressing again. Your DNA repair machinery comes back online. Honestly, when I read this in a 2014 review published in the Journal of Experimental Medicine by Boltman at the University of North Carolina, what stopped me was a comparison in the discussion section. The HDAC inhibiting mechanism of butyrate is pharmacologically parallel to vorinostat, sold as Solinsa, FDA approved for certain cancers, at a monthly cost running into thousands of dollars. Your gut bacteria make a functional version of this molecule when you feed them resistant starch. In plain terms, the FDA approves drugs targeting this pathway because HDAC inhibition is a validated cancer strategy. Faecalibacterium prausnitzii does it every day when given the right substrate. The broccoli piece connects directly. Sulforaphane activates the NRF2 pathway, regulating a separate set of DNA repair and antioxidant genes. Butyrate opens the chromatin through HDAC inhibition. When both operate simultaneously, the genes both pathways need are more accessible than either achieves alone. Resistant starch and broccoli together create a dual epigenetic protection system in your colon. That combination is not an accident of the protocol. A 2019 trial in cancer prevention research by Zhu and colleagues had 25 adults eat 2 ounces of walnuts daily for 2 to 3 weeks, then analyzed colonic biopsies. Gene expression shifted toward upregulation of butyrate-related metabolic pathways and downregulation of cancer-promoting genes. Translation: walnuts, through their combination of elagitannins and omega-3 fatty acids, extended the same epigenetic logic across a different molecular pathway. Robert, 64, had been avoiding potatoes for 3 years because someone told him they spike blood sugar. True, for hot, freshly cooked potatoes. But cooled? A different food metabolically. He started making a cold rice and lentil bowl with broccoli 4 days a week. 7 weeks in, he described his digestion as better than it had been in his 40s. His gastroenterologist attributed it to less processed food overall. Robert nodded and didn't correct him. Now, the mistake that destroys all of this before it matters. Buy raw unmodified potato starch. Bob's Red Meal is widely available. Start with 1 teaspoon in cold water or yogurt. Never heat it. Build to 2 tablespoons daily over 4 to 6 weeks. This single addition provides 8 to 16 grams of resistant starch per day without restructuring your meals, and it is functionally the closest food equivalent to what the box trial used. Newcastle University's box trial, led by Dr. John Burns' research group, enrolled 918 patients over 4 years of intervention with 10 years of follow-up, the largest randomized controlled trial of resistant starch in history. The long-term analysis published in Cancer Prevention Research in 2023 found a 60% reduction in cancers of the upper gastrointestinal tract among patients who received the resistant starch supplement. The protective effect emerged after the intervention period ended, suggesting butyrate had made durable epigenetic changes, not temporary, lasting. But here's what people don't realize they're doing. You refrigerated potatoes last night. The RS3 has formed. You microwave them on high for 2 minutes. You've converted the majority of RS3 back into digestible starch. The cooling built the structure, the high heat reverses it. Gentle warmth, or eaten cold, preserves most of the RS3. Steaming hot undoes the work entirely. The banana parallel. Green gives you 6 to 9 grams of resistant starch. Fully ripe with brown spots, under half a gram. Every day of ripening converts RS2 to sugar. Keep bananas at different ripeness stages so you always have a green one available. The psyllium husk most people use as their fiber supplement ferments differently, feeds different species, and does not efficiently amplify the bacteria that make butyrate. Psyllium has real cardiovascular benefits, but it is not a resistant starch substitute for this mechanism. Before the full picture, a direct note to anyone on blood sugar medication or anticoagulants like warfarin. Resistant starch significantly reduces postprandial glucose. Medication doses may need adjustment as your diet shifts. Work with your physician. If you're on warfarin, significant dietary changes warrant an INR check. This protocol complements medical supervision. It doesn't replace it. Butyrate works through 3 simultaneous mechanisms. The first, selective starvation of cancer cells through the Warburg effect, as we covered. The second, HDAC inhibition that reopens tumor suppressor and DNA repair genes cancer have been silencing. The third, the one most overlooked, your colonocytes replace themselves every 3 to 5 days, one of the fastest cell turnover rates in the body, intentionally. Cells accumulating genetic damage get shed before they can proliferate. Butyrate drives that renewal rate. When butyrate is deficient, turnover slows, damaged cells persist, and mutations compound. Butyrate also maintains the tight junctions between colonocytes, the seals preventing bacterial products from leaking into the bloodstream. When those fail, systemic inflammation rises. And systemic inflammation is a co-driver of cancer progression, cardiovascular disease, and accelerated aging simultaneously. The colon wall's integrity is not a local issue. Honestly, the studies don't have a perfectly clean answer for why resistant starch doesn't work equally for everyone. In the University of Michigan trial, roughly 30% of participants showed minimal butyrate response. The most likely explanation, severe depletion of butyrate-producing bacteria through years of antibiotics and low-fiber eating, means the fermentation capacity isn't there yet. Adding fermented foods, plain yogurt, kefir, unpasteurized kimchi, refrigerated sauerkraut, alongside the RS protocol may help seed the bacterial environment RS feeds. The timeline may extend to 8 to 12 weeks, rather than 3 to 6. That is not a reason to stop. The honest caveat on the box trial, it was conducted in Lynch syndrome patients, people with inherited genetic mutations dramatically elevating cancer risk. The mechanistic data, Warburg effect, HDAC inhibition, operates in all human colons. The observational epidemiology, across diverse populations, strongly supports high-fiber diets for colorectal cancer prevention generally. But a large randomized trial of resistant starch, specifically in average-risk populations, hasn't been completed with cancer as the primary endpoint. The mechanistic case is solid. The direct causal chain in general population cancer prevention remains partly inferential. You should know that distinction. What to stop? Reheating cooled starches to steaming hot. treating psyllium as a resistant starch substitute, letting bananas fully ripen if RS content is the goal, and stop treating colon health as a passive waiting process. The Africa study says clearly that your colon's cancer risk profile is being ridden by what you eat, in a time frame measured in weeks. Timeline. Week 1 to 2, expect bloating as your microbiome shifts. By month 1, bowel regularity typically improves. Butyrate directly regulates colon motility. By months 2 to 3, butyrate-producing bacterial populations should be measurably closer to optimal. Mucosal inflammation declining. Epigenetic DNA repair mechanisms more consistently active. This is a long-term protocol. The box data showed protective effects increasing after the intervention ended. The changes are cumulative when the diet is sustained. Now, the gun planted at the start. Frank's doctor said, "Come back in 3 years." Frank heard that as monitoring. The biology says something different. 3 years of cellular momentum going somewhere. If his colon is continuously supplied with adequate butyrate, if mucosal renewal is running at its proper rate, tumor suppressor genes staying open through HDAC inhibition, precancerous cells being forced toward apoptosis daily, then 3 years is a meaningful window of protection. If he does nothing different, if the psyllium goes back in the cabinet and the potatoes keep coming out of the microwave steaming hot, then 3 years is time his gastroenterologist will need to account for at the next colonoscopy. The advice was right. The specificity was missing. Eat more fiber becomes actionable when you know which fiber, how it's prepared, and what it does that nothing else replicates. The daily protocol. 1 to 2 medium potatoes cooked the evening before, refrigerated, eaten cold or gently warmed the next day. 1 green banana blended into a morning smoothie with 2 tablespoons of raw ground flaxseed. Half a cup of lentils, white beans, or chickpeas most days. Lightly steamed broccoli 4 or more times per week. Chopped, rested 10 minutes, steamed briefly. 2 cups of green tea daily steeped at around 165 degrees Fahrenheit. And the backup for difficult days. 1 to 2 tablespoons of raw potato starch stirred into something cold. Never heated. That's the non-negotiable. The preparation is the intervention. A cooled potato is not the same food as a hot one. The biology doesn't round up for convenience. If you're seeing blood in your stool, experiencing unexplained weight loss, or noticing a change in bowel habits persisting more than 6 weeks, stop watching and call your doctor. Those are not resistant starch questions. For everyone else, the research is consistent, the mechanism is well characterized, and the intervention costs potatoes, green bananas, and a bag of lentils. The complexity isn't finding the food. It's remembering that dinner tonight needs to be cold by tomorrow morning. If this changed how you think about your next meal, share it with someone who needs to hear it. There's a related video on the specific gut bacteria that predict colorectal cancer risk years before any symptom appears, and what the research shows about rebuilding them. I'll see you there. Keep investigating. Dr. Becker.